actually come from a background of infectious diseases medicine and had worked quite extensively both here at Brigham and Women's Hospital, Dana-Farber, and then most recently at the Mass General in the area of transplant infectious diseases, where we really are involved in patients with incompetent immune systems because of what we've done to the patients in order to accept the transplant. But there's a tremendous overlap, as you can imagine, between infectious diseases and cancer, which is important to understand, not least from the point of view of the role of vaccines and our understanding of the interplay between the immune system and, in the case of infectious diseases, the pathogen, but in the case here of the cancer and the immune system. And I just put up this slide, and it's by no means comprehensive, but you could draw the same thing up and put in red any particular intracellular pathogen on the red, in the red circle. Here we've put cancer, and on the green we've put the host, and there is an incredible interaction going on. You can call it war and counter-war, offensive and counter-offensive, between the immune system that's trying to eradicate the abnormal cell or infected cell, and the pathogen or cancer on the other side launching a counterattack based on its own biology. And I think that the remarkable technologies that are now available to look at this have raised the whole field of personalized cancer vaccine development and personalized understanding of the individual's immune response to either a cancer or an intracellular pathogen to a much higher level. And for that reason, I just wanted to show this slide. This is a new technology called CyTOF, which is called mass cytometry, where you can look at the entire immune and inflammatory system of an individual with about 500 microliters of blood, and you can do it over, obviously with that type of volume, you can do it over again and again and again over time, and monitor, and I don't know, there is a pointer, right? There's some sort of laser pointer here. I love this one because it has this right-hand corner thing which says danger, like as if you press that, you could finish your presentation very rapidly. But basically, this is a representation of the clusters of different cells in a 500 microliter drop of blood divided by mass cytometry, where you can identify 40 different markers simultaneously, and then using computational algorithms cluster them into different subpopulations of cells, whether it's T cells, macrophages, dendritic cells, and so forth. And the minute I personally saw this type of image, as opposed to focusing entirely on T cells or what the B cells are doing or the dendritic cells, was that you realize the amazing complexity of the interactions that must be occurring in your blood or in any tissue when all of these cells are present or present in different proportions and capable of interacting with each other. The second image, so this is the clustering of CD8 T cells and CD4 T cells in, again, a 500 microliters. So now, previously you saw the phenotypic clusters. Now these are functional clusters of at least 1, 2, 3, 4, 5, 6, 7, 8, 9 different phenotypically different subpopulations identified by the clustering algorithm of CD4 T cells in the blood, and similarly a significant number of CD8 subpopulations just sitting there in the blood. So again, a simplistic approach is just not possible in this context. We have to understand the diversity of the potential immune reaction against cancer and intracellular pathogens. Now we talk about a cancer vaccine renaissance, but obviously from my point of view as an infectious diseases physician and scientist, vaccines are the most tremendous and impactful healthcare intervention that we have. And it's an absolute tragedy to me that we live in times where the contrary can be stated as strongly as what I just stated about the importance of vaccines. But regardless of that, vaccines in the infectious diseases context have been extremely effective in preventing disease. And obviously, we know about the first cancer vaccines and the increasing number of immune checkpoints that potentially could be used in conjunction with vaccines, so releasing the immune response to the cancer with the checkpoint inhibitor and then targeting it to specific tumor-specific antigens or neoantigens is obviously there. And then we also are aware of the several thousand vaccine and immunotherapy clinical trials that are ongoing currently in the US and the EU. So I'm trying to line up the complexity of the immunology that's going on and then the complexity of the actual practical clinical studies that are going on. And how do we rationalize all of that and actually do combination or targeted vaccine studies and design targeted vaccines in a rational way? There are also very important lessons from vaccines to intracellular pathogens. Okay, we sit in a center here that has worked for over 30 years on the HIV vaccine, and that we think that we're very close to one now, but we have to think of the 30 years of investment in trying to identify a vaccine for a pathogen that has 9,000 base pairs and is capable of manipulating the immune system and damaging the immune system in order to foster its own existence enormously successfully. Now contrast that with a cancer cell with the entire human genome at its available to itself in a sense as an adaptive system to keep the immune system at bay. So we have to have a certain humility around the way that we design vaccines or personalized vaccines for cancer by understanding the biology both of, in this case, the target, which is the cancer cell, and the diversity of the immune system that's up against it. It's not a minor point that it has taken 30 years of research and billions of dollars, and we still don't have an effective HIV vaccine because of the complexity of those two aspects of the system. So we come into cancer vaccine development with that knowledge. Now, obviously, traditionally, there are what are known as tumor-specific antigens that have been detected over many years, that have been present, found to be present selectively, or mostly selectively, on specific types of cancers, whether it's CA125 and CEA on ovarian cancer and so forth. But they could serve and have served as immune targets for vaccination. And the other thing is the issues regarding the fact that when you start targeting these types of antigens, there's the potential to induce tolerance. We have an immune system that if you keep hammering it with the same, alerting it to the same antigen, ultimately develops tolerance. So these issues we know arise when you vaccinate individuals. And also, I think another remarkable aspect of the immune system, which is, again, it sort of goes through waves, but it's again extremely interesting to scientists at the moment, is the fact that the minute you turn on an immune system to do something, there are forces within your immune system that are activated to switch it off. And we understand that as clinicians, that the so-called overzealous inflammatory reaction, the overzealous cytokine storm and so forth is not good for your health. And you do have naturally occurring breaks that are beginning to be applied even as you accelerate your immune response to a targeted antigen. And that's a very important component that we have to be aware of when we're trying to tune the immune system with a vaccine to upregulate its immune reactivity to a specific cancer antigen. The other targets, which may have been talked about previously, but obviously neoantigens that are expressed on tumors. So given the instability of the genetics of cancer cells, new antigens are being expressed by these cells that can be potential targets for the immune system. And in fact, even in native settings, you will start to generate immunity against specific neoantigens. So the fact that the genetics of the cancer cell are unstable relate to the fact that mutant proteins are basically being expressed. And Mark Cobbold, in addition to this, has shown differences in the phosphorylation of proteins. So you can imagine the levels of targeting that could be brought in here, both on tumor-associated antigens and neoantigens and modifications of those neoantigens that could be targets for a cancer vaccine. And I think the key thing that's been shown so far in both preclinical and clinical studies, that if you incorporate these sequences of neoantigens in vaccines, and we'll get onto the design of some of these, you can actually evoke potent CD8 T-cell responses to those antigens. Now, whether that then results in an effective immune response in terms of controlling the tumor raises as big a question as it is in HIV, where you can raise significant peripheral CD8 responses to HIV and still not control the disease. So that balance is still out there to be definitively shown, that this link could actually lead to significant impact on tumor progression. And these are just some of the neoantigen vaccines under exploration currently. They basically consist on screening the tumor cell for the presence of neoantigen vaccines, distilling them down, and I'll show some of the details of that, into targetable peptides that could be included in a specific vaccine. And these are sort of you get the tumor sample, you have algorithms that go from DNA and RNA sequence to identify the mutations. You do HLA typing that allow you to predict specific peptides that the immune system of that individual should actually raise an immune response against. You can prepare each one personalized to that individual patient, and then obviously deliver it with some sort of strong adjuvant or a checkpoint inhibitor to the patient. And this is the basis of clinical trials in neoantigen or personalized cancer vaccines. And I think stressing here is absolutely akin to combination chemotherapy or combination approach to combination drug therapy in diseases, is that we recognize that a vaccine alone in this context, given the armamentarium of the cancer cell to work out ways around whatever you do, that you will need to use it in combination with both something that adjuvants the immune response in a general sense, like a chemical adjuvant, and in a more targeted way, but not targeted to the antigen, with a checkpoint inhibitor. And just to highlight to you that there are several companies and several academic groups that have developed ways of going from sequence to identifying specific peptides that would evoke an immune response, and it's been developed into a fine art in the T-cell epitope finding area, and there's a number of selection processes to make sure that you're raising an immune response in your vaccine to a peptide that is not going to cross-react with a self-antigen, or potentially even your microbiome or other aspects of your native peptide content, as it were, native immunological peptide content. You can look at relative TCR, T-cell receptor affinity, clonal specificity, and obviously, as I said before, the self-reactivity that you do not want to induce as the result of giving a neoantigen vaccine. There's room for improvement, obviously, like with all of medicine. It's always striking to me that we're never quite there, and I've often been told that's why we're always practicing medicine as opposed to actually doing anything that is complete. So at the moment, things are based predominantly on DNA or some form of sequencing. Sequences may be cross-reactive, and we need to know how to optimize these sequences for actual antigenic presentation, TCR fit. The other point that we've begun to work on in the team, with the team at a large government computation lab, is identifying B-cell epitopes. The usual thing in science is trends that we focus on at the T cell. That's where we want to get the activity, and other groups may look at another cell. But I do think that the future of vaccine is obviously to evoke a broad immune response, whether it's engaging dendritic cells in immune activation, T cells in targeted cell-mediated immunity, and potentially B cells, which have been left out of this somewhat in terms of targeting with antibodies. The other thing from a clinical, I think just on my last slide on the other issue is obviously adjuvant delivery systems are not optimal. We've got some pretty gross adjuvants out there that are not good, and we've also, studies have been shown that depot-based adjuvants that stay in the site where you vaccinate cause a retention of the immune cells that you want to go after the tumor, but they congregate at where you vaccinated the patient as opposed to in the tumor. And the vaccines themselves cause significant, the adjuvants cause significant side effects and reactogenic in their own right. The other issue is the cost, and this was just an assessment one of the MBAs who are associated with our team put together. If we treated everyone in the US with prostate cancer with a vaccine or immunotherapy, the cost is $10 billion. So at the current pricing of these complex products, we're facing the potential, as we know, we're regularly told that the healthcare system will bankrupt the nation, but it is something that we have to be aware of, that ultimately the personalized vaccines would have to be appropriately costed in order to be broadly applicable. And I just really came in at the last kind of question period about, the fact that we want to get the science and medicine out into the community. How do we do this? How can a patient living 20 miles outside of Boston, going to Norwood Hospital, to give an example, get access to this? Well, if we do that, what are the cost implications both for that hospital and for the system in general if we start to do that? I just show you one of our products which self-assembles a dendritic cell immune activating protein, which I'm not going to bore you with because it's just pure nerdy science, but it's interestingly a MTB, a Mycobacterium tuberculosis-based protein that, go figure, activates your immune system very broadly, and TB encodes that. The current paradigm would be, why is MTB activating your immune system? It actually activates parts of the immune system that allow it to use that part of the immune system as a host for its own infection. And then we couple that with either with biotinylated peptides sorted from both neoantigens and tumor-specific antigens in order to generate a vaccine. And the bottom line of this is that we have contemplated a system that would go from tumor, and we've been obviously doing preclinical studies, to interestingly, a plant-expressed fusion protein between MTB HSP70 and the peptide strings to create a vaccine within about 19 to 20 days for delivery. And this system here, which is a revamp of the whole nicotinia or tobacco industry into a recombinant GMP protein manufacturing system, has reduced the cost of those fusion proteins. So we could deliver this potentially at much lower cost in 20 days. And this slide also epitomizes one of the other challenges, that when you've identified the patient you want to give the vaccine to, you need to get that vaccine as fast as possible and before the tumor and the immune system dynamically change the environment. And so we reckoned 20 days would be at our first shot what we would need to go for. Here are just a summary of the issues that we came across and ended up looking at plant-based systems, which are rapid production. You can essentially produce a rack of plants in about 8 days, producing enough protein for an individual patient. So that would be, that's sort of the comparison between solid state and a plant-based system for expression. I just don't want to go over time here. These are some of the key things, and I related this before, the 9,000 base pair genome versus the 3,000 megabase genome, and what the repertoire of the cancer cell has to utilize to evade the system. And then some aspects of the future related to the fact that we have these thousands of immunotherapy combinations going on. We've got millions of patients, continuous new data feed. How do we understand what this data is telling us in order to identify which patient will benefit from which drug, which patient will not benefit from which drug? And I was struck recently, interested by a patient in my network had been diagnosed with melanoma, and this was maybe a year ago, and been told, well, we could start immunotherapy, but there's a 1% chance that you'll have a potentially lethal consequence of that immunotherapy. So the patient weighing up, okay, they feel perfectly well and they've got a localized melanoma lesion. If they have immunotherapy, there's a 1% risk. These are the types of things where understanding the likelihood of a patient benefiting before you give the therapy is going to be helpful. And then I really think we have to think about controlling costs. We are in a situation where, you know, the general tendency is that the drug that is developed costs more than the previous generation of drugs. And at the moment, in the context of cancer vaccines and cancer immunotherapy, that is entering not just a doubling phase, but that's getting much more than that in terms of what the cost is. And I think we have to be aware of that. I just want to, if I've got a few more minutes, just to talk about where we're going with addressing what patient will benefit from what cancer immunotherapy or cancer vaccine, how to target that cancer vaccine and immunotherapy, and also then look for indicators of efficacy or even predictors of efficacy, so that we are not giving all patients the same thing in order to find that 10 to 18% of them respond. We actually identify the 10 to 18% of responders before we actually start, or potential responders before we actually start. And even if we could reduce it to the patient population that's treated from 100% to 50%, I think that would be a benefit. So we've been looking at systems with a company called BostonGene. We're working in scientific academic collaboration with them to see if we can determine ways to actually profile the entire tumor, both the immune response, the tumor itself, the mutational burden, and so forth, and then integrate that data to provide insights to the clinician with regards to clinical decision making. And in our first study, we've been looking at both RNAseq based studies and this mass cytometry CyTOF analysis of non-small cell lung cancer. Just the key first step was just to cross-validate that if you looked at the RNAseq algorithm that was based on specific subpopulations of immune cells being profiled from an RNA sequencing point of view and identifying the specific RNA signatures of those immune cells, whether when you come up with that algorithm to define different subtypes of immune cells, you can correlate it with the actual immunophenotype of the cells generated from the tumor. And this shows the types of correlations you're able to see by looking at RNA-seq and simple flow cytometry. When we looked at mass cytometry, that is the RNA-seq being run through an algorithm to define what immune cells you have in that mix, compared to the phenotypic studies that actually positively identifies an antibody on a binding to a protein on a cell, we actually got very good correlation. So these predictive algorithms are actually very good at defining the immune milieu that's present in the tumor. And then if you look at the tumor itself, and we've been looking at specific tumors, with regards to the comparison of that tumor RNA-seq profile and the TCGA database, you can actually segregate the tumors that we're testing, and they're either falling into an adenocarcinoma or a squamous cell carcinoma, but based on their RNA-seq profile, with the ultimate goal of generating a full sort of portrait of the tumor, including, it's difficult to actually see both screens here, but including the components of the immune system, the stromal components, and the tumor itself, identifying the mutational burden, the target gene expression regarding the tumor's expression, the tumor microenvironment, the stromal, the immune cells, and so forth, in a programmatic way where one could actually define that individual tumor and their immune targets within that tumor simultaneously in order to potentially guide clinical decision making. This is very much at a prototype level and has not been fully tested, but we're working with the thoracic oncologists and thoracic surgeons at Mass General to develop this and see whether there is actually any legs to this with regarding to guiding clinical decision making. The other thing I want to talk about, and we do live in a remarkable time, and there's an explosion in technologies that allow us to look more deeply at cells, basically. And we would be utterly remiss if in this age of developing personalized vaccines, we don't exploit these technologies to the absolute full. You know, what this offers to us in terms of understanding tumor biology is very significant. I'm sorry, it seems to have moved up here, but the bottom line is there's ways of actually looking at tumor samples that go way, way beyond immunohistochemistry, where you could look at 2, 3, maybe 4, if you're lucky, particular labels. And the technologies that are available are mass spectrometry, imaging mass cytometry, which is similar to the flow-based or mass spec-based mass cytometry, where you can actually look in the tumor sample for 40 different markers simultaneously, and then using computational algorithms, pick out those 40 different proteins where they overlap on any individual cell in your tissue sample. I haven't got our latest work here presented on prostate cancer, but the images are absolutely stunning, because you can now look at 40 different proteins. We could look at immune cells, tumor-specific markers, and so forth, and get a geographical understanding, both pre- and post-treatment, of the location of the immune system and the location of the tumor samples. And here we have merged imaging in the context of lung tumors that we are doing. These are just, you would highlight in this merged image maybe 3 or 4 of those antibodies that have been identified. Nearly done. And then mass spectrometry imaging, that allows you to take a slide, take a fixed frozen paraffin embedded slide, and scan it with a laser at a resolution of about 5 to 10 microns every single spot, and do mass spec on the vapor that's coming off at that spot, and then do using algorithms, define the chemical composition from lipids all the way to proteins at that type of resolution on your slide, where every single molecule has a defined mass that you can go back and identify precisely what it is, and then recreate it on the basis of cellular structure. I mean, this is mind-blowing technology, but it allows us to go much more deeply into defining the individual person's tumor, the targets that are on it, and then if we treat it, what is happening to the immune system in response to this. And I just point this up as just remarkable technology that we're just beginning to get into. And there is a group in London that are using this technology at the operating room side when tumors are being removed, and they're doing real-time mass spec on the margins to define whether the brain tumor has been fully resected or not. It's quite remarkable. So just to say big thanks to many collaborators in the work that I've presented that we're doing, particularly Patrick Reeves and Roxandra Serbalescu, the senior scientists in my team, have brought this technology to VIC, the center that I direct. And then, you know, the BostonGene, the company that's working on the algorithms for defining the tumor and the immune system within that tumor, and then other collaborators that have helped support the science and technology of this particular work. Thank you very much indeed. I'd be happy to answer questions.
Yeah, thank you, Mark. That was great. It really is, it does feel like living in a sweet shop these days as an immunologist. There is so many cool things you could do. Right, I'm mindful of time, but I'm just wondering if there is one question that we could field to you. You stunned your audience.
Stunned them.
Too much? No, not one? I'm going to ask a question then.
Uh-oh.
Yeah, vaccination has been like one of the most impactful developments in medicine, and history teaches that it's actually been very cost effective as well in terms of preventing disease. In terms of the emerging vaccines for cancer, they seem to be somewhat complex. You've often got this heterologous sort of prime boost requirements, right. RNA. You've actually got quite sophisticated technologies. How do you think the cancer vaccine field is going to impact on other vaccination strategies?
Well, beyond cancer, obviously, I mean, there's a very interesting story with regards to the neoantigens that are expressed in type 1 diabetes and why people raised an immune response against those neoantigens that cause type 1 diabetes. So where cancer vaccines and this edge between immune activation and immune regulation as manipulated by a vaccine tilts more to immune regulation for vaccines in the context of autoimmune disease. So what we're learning about cancer vaccines, where the goal is to activate the immune system, then we learn that actually there's some things that vaccines in cancer do that actually end up with a regulatory response. Can we use that component in the context of autoimmunity? Very, very interesting.
Great. Well, thank you very much.